Hi students, welcome to Ligase Academy. In our previous video, we already studied about prokaryotic transcription. In this video, we are focusing on eukaryotic transcription, especially the structure of RNA polymerase and the components of promoter elements. As we know that eukaryotic cell has a nucleus, so it separates the process of transcription and translation. Transcription occurs within the nucleus and translation occurs within cytoplasm. And this eukaryotic transcription proceeds in three sequential steps, that is initiation, elongation and termination. In case of prokaryotic RNA polymerase, that initiate the transcription of different types of RNA. But in case of eukaryotes, RNA polymerase comes in three variations. Each RNA polymerase translating a different types of genes. There are three specific RNA polymerases and that transcribing different types of genes. The given table shows the different types of RNA polymerase and the different types of RNA that synthesize. First one, RNA polymerase 1, that initiate the synthesis of ribosomal RNA, that is 28S RNA, rRNA, 18S rRNA, 5.8S rRNA. The location of this polymerase is in nucleolus. Then second type, RNA polymerase 2. This enzyme involved in the synthesis of messenger RNA, small interfering RNA and small nuclear RNA. Location in nucleus. Then the third RNA polymerase 3. It involved in the transcription of gene that code for tRNA, 5S ribosomal RNA, snRNA U6, signal recognition particle RNA. We are discussing about transcription using RNA polymerase 2. RNA polymerase 2 known as RNAP2 and POL2. It is one of the three RNA polymerase enzymes found in the nucleus of eukaryotic cells. It is a multi-protein complex because it has multi-subunit and this protein having analogous subunit to that of a bacterial RNA polymerase. It transcribes DNA into precursors of messenger RNA, small nuclear RNA and microRNA. It has approximately 10 to 12 subunits. These subunits are represented in RPB 1 to 12 format. Among the 12 subunits in RNA polymerase 2, there are two large subunits that are known as RPB1 and RPB2. RPB1 is the largest subunit and it is analogous to beta-clamp of RNA polymerase in prokaryotes. And they contain a catalytic motif and it also contains a clamp domain that can assume an open state in the absence of RNA in the active site or a closed conformation in the presence of RNA-DNA hybrid. Clamp increases the processivity of RPB1. RPB1 contains a large carboxyl terminal domain that is involved in different stages of transcription. This carboxyl terminal domain contains a heptade repeat with a repeating number varying from 20 to more than 50. These heptade repeats are tyrosine, serine, proline, threonine, serine, proline, serine in this order. Among them, the second and the fifth serine undergo phosphorylation. These are the repeats. These are the minimum repeats that are required for viability. We know that RNA polymerase 1 and RNA polymerase 3 that don't have this carboxyl terminal domain. In RNA polymerase 2, this heptad repeat, serine and tyrosine in the carboxyl terminal domain are phosphorylated. Before initiation, these are in unphosphorylated state. During initiation, first phosphorylation occurs on 5th position serine, and it acts as a docking site for elongation factor and capping enzymes. The phosphorylation on 2nd position serine correlate with promoter escape. Splicing enzymes, poly-A tail enzymes, and protein required for terminations are also associated with this 2nd position serine. Then we are discussing about the promoter regions for the enzyme RNA polymerase 2. Multiple sites are required for transcription initiation. So these transcription control regions are further divided into two. One is core promoter and the other one is regulatory promoter. Core promoter is the specific region in DNA where RNA polymerase is recruited. It contains data box, initiator, CpG island, motif 10 element, downstream promoter element, downstream core element, transcription factor 2b recognition element. The next portion is regulatory promoter. As the name indicates, it regulates the transcription at core promoter. It has enhancer, promoter proximal element, insulators and silencers. No box in prokaryotes. Then we are discussing about the important part, CpG island. Generally in eukaryote, where cytosine is methylated, it has less GC content in the genome than the expected frequency. Deamination of methyl cytosine forms thymine. It leads to GC to AT transition. Less GC content because of this GC to AT transition. But there is some isolated pockets where cytosine is hypomethylated. These regions are known as CpG island. GC contents are normal in this region, and these CpG islands are found in the housekeeping genes. These CpG islands don't have a precise binding site for RNA polymerase enzyme because it is a site for constant transcription. General transcription factors bound in this region prevent methylation. It contains origin of replication, so nucleotide turnover in these regions are high, which decreases the chance of methylation. Also, core promoter element found downstream of transcription start site. These elements include downstream promoter element, motif 10 element, downstream core element. Then we are discussing the downstream promoter element. It is located about 13 nucleotide downstream of transcription start site. It is a GC-rich sequence and it is functionally analogous to Tata box. It is found in Tata-less promoter and it functions cooperatively with the initiator for the binding of transcription factor 2D in the absence of Tata box. The TF2B recognition element, it is known as B recognition element and expressed as BRE. It is a cis regulatory element that is found immediately near Tata box. It consists of 7 nucleotides. There are 2 sets of BRE. One is located upstream and other one is located downstream. The upstream located is known as BREU. It is located immediately upstream of Tata box as seen in the picture. Then the next one located downstream, it is known as BRED. It is found 7 nucleotide downstream. Then the downstream core element, it is represented as DCE. It is found in between +6 and +24. It is discovered in beta globin gene. Motif 10 element MTE. It is located downstream of transcription start site and it, as in the picture, it is found just upstream of downstream promoter element. It is a 10 nucleotide long sequence and it is cooperatively interact with initiator to stimulate transcription. MTE can act in synergy with either Tata box or DPE, that is downstream promoter element. MTE contribute to TF2D binding to core promoter. Then we are discussing about the concept supercore promoter. It is known as strongest promoter. This supercore promoter consists of a Tata box, motif 10 element, initiator and downstream promoter element. The transcription factor, that is transcription factor 2D, shows high affinity to this supercore promoter. This directs the high amount of transcription by enzymes RNA polymerase 2. It is observed in metazoans. We are discussing about the regulatory promoter element. As the name indicates, it regulates the promoter. regulatory promoter element consists of proximal element, enhancers and silencers. First one, promoter proximal element. Promoter proximal elements are located proximal to the core promoter, either upstream or downstream. It is 6 to 10 base pair long. They recruit transcription factors called activators. These activators interact with the co-activators. Co-activators increases the accessibility of core promoter to initiation factors. This proximal promoter element is orientation independent, that they recruit co-activators either in the direct or inverted positions. They are distance dependent, that deletion of 20 base pair or less spaces between the proximal element and the core promoter has no effect in the transcriptional efficiency. The examples for proximal elements are GC box and CAT box. Enhancer. Enhancer functions similar to promoter proximal element. They are located approximately 1000 nucleotide away from core promoter. It is 200 base pair long. This enhancer sequence binds with activators and form a complex called enhanceosome, and it is orientation and distance independent, and it stimulates transcription with the help of activators. Silencers. As the name indicates, it silences the promoter, that it represses the transcription. It functions analogous to enhancers and antagonistic to also. They are orientation and distance independent. They recruit repressors. These repressors interact with the co-repressors, and this region will be transcriptionally silent with heterochromatinization. These silencers repress transcription in two ways. That one, modify and remodel the chromatin. The second way is preventing accessibility of the enhancers to activators. The final regulatory promoter element is insulators. Insulators are specific DNA sequences that recruit proteins which prevent the spread of heterochromatinization and euchromatinization. There are two types of insulators: enhancer blocking insulators and barrier insulators. Enhancer blocking insulators, as the name indicates, it blocking the activity of enhancer by preventing the spread of euchromatinization. And the second one, barrier insulators, act as a barrier. It prevents the spread of heterochromatinization to adjacent region and preventing the enhancers and silencers from regulating neighboring genes. Hope you all understood this video. We will continue eukaryotic transcription stages in our next video. Please stay tuned. If you like this video, please like, share and subscribe YouTube channel like Csir Academy, your educational catalyst.